Thank you, Ramon, for the introduction, and thanks to the organizer for the invitation. I will concentrate in 4 topics related with the cell therapy of diabetes and the complications, and that's the title, but the subtitle could be From Bench to Bedside and Back to the Bench. And I will show at the end of the lecture an example of how learning from a clinical trial allows you to go back into the lab, ameliorate the treatment, and come. And in each case of the 4 groups, I will comment how far we are from the clinical applications. And this is the last one, so even if it's tedious and boring, I will try to make shorter the presentation. And basically, this is a work done inside CIBERDEM with Dr. Fran Martien, Karim Dacha, Anabel Rojas, Benoit Gautier, Francisco Bedoya, and Juan Tejedo in CABIMER, the Andalusian Center for Molecular Biology and Regenerative Medicine. A few years ago, well, exactly 10 years ago, we met in Düsseldorf, and that was Dieter Kabelitz, an immunologist, asked to a group of fellows, I was one of them, just in case we have to find a solution for diabetes, what do we have to solve? And the summary of 2 days of discussion is this publication in which we finally say that if we want to have a treatment for type 1 diabetes, we will need to solve cell replacement and induce the tolerance. So that's the 2 main questions at that moment we should solve, and I will go through these aspects. So, in fact, the presentation is organized in 4 topics: immunomodulation, replacement or substitution, regeneration, and the treatment of complications. Immunomodulation is a big topic in which there are a long list, probably 20 clinical trials in phase 1 using monoclonal antibodies that never went into phase 2. So very expensive, very clear the rationale. They work with the animal models, but we couldn't follow with that. And we tried to approach a few years ago asking whether we could use cells to immunomodulate. And that was not completely original idea, because it was previously published that mesenchymal stromal cells are immunomodulators. They block T-cell proliferation, decrease TNF-alpha and interferon gamma, etc., increase interleukin-10, etc., etc. And even there are some preliminary preclinical data in which it was seen that mesenchymal stromal cells are good in islet transplantation in non-human primates. There is also some published data, but not from a trial. And there is also a trial from Osiris. Prochymal is bone marrow mesenchymal stem cells used for type 1 diabetes mellitus, and some data published not from well-organized clinical trials. So there's some previous information, and what we wanted to see, could we have a better cell or even a better way of administrating the cell? These results I'm going to present have not been published, and the idea is if we have another model that reproduces our idea of type 1 diabetes, and the idea is that type 1 begins with a strong inflammatory response. So the first stage should be to stop the inflammation in the islet, which creates the insulitis. For doing that, we use a model of type 1, which is the RIP mice, in which the transgenic mice co-express, in the beta cell, express the co-stimulatory molecule for T cell, and that generates a strong inflammatory response, the insulitis. It's very clear. You can see these are control animals. These animals immunized. Well, the way that you induce type 1 diabetes in this model is that you immunize the animals with preproinsulin, and that induces a strong insulitis, the lymphocyte attack, and obviously an increase in the blood glucose. So we develop new cells. They basically belong to the family of mesenchymal stromal cells that, when not applied to these animals, decrease the response in the islet. The insulitis is small. That will correspond to this level of glucose, but with double the glucose or the same, double the dose or the same dose twice, we normalize the blood glucose in these animals. So we have extended all the results about the duration of the effect, the need for a new dose when 7 or 8 weeks after the transplantation, the dose that we need. So not only the dose, but the protocol and the way, but what is more important, well, you could see if these are the control animals, these are the non-transplanted animals, and the transplanted animals exactly reproduce the situation. See that you have to compare this transplanted animal with animals that are type 1 diabetes, will generate this insulitis and lose the beta cell population, or almost lose the beta cell population. And the effect is dose dependent, which is important for any therapy. You have to demonstrate that the effect is cell dependent. And perhaps summarizing a long list of experiments, perhaps the nicer observation is, if we consider this is more or less the immune cell profile in control animals, that will be the cell profile that includes T cells, CD4, CD8 positive cells, B cells or macrophages, and only visually you could see how we normalize the profile of immune cells in the pancreas of the animals. So now we know the process by which the changes in the lymphatic nodes and in the peritoneum generate all this process. So this group of experiments, we think we have enough preclinical data because that using mesenchymal stem cells derived from umbilical cord, but we have also the same set of experiments with adipose derived mesenchymal stem cells under certain conditions of expansion, media, etc. So we think we have data to go into a clinical trial. Why I am saying that? Because we have previous data from the group of Karolinska, from Ole Kosgren, who is collaborating with us as well, by which they use bone marrow mesenchymal stem cells in adult diabetic patients. So we will like to use this kind of cells, adipose-derived cells, autologous from the same patient, in patients which are, who are recently diagnosed from type 1 diabetes. In the case of Spain, which are more than 18, so adult patients, recently diagnosed, and it's quite easy to organize this trial because we have this cellular medicament has been approved. It's approved in my center, in our GMP facility, so we have the production, we have the logistics for transport the cells everywhere, and the implantation is very simple. So basically the endpoint will be, well, for a pilot study should be safety and feasibility, but at the same time you could measure the C-peptide. The patient should have a certain level of C-peptide and see whether C-peptide is kept. So we are close to a trial that, in my idea, should be done inside CIBERDEM and with the interaction between basic scientists and clinical scientists in CIBERDEM. The second topic I want to discuss is about replacement, substitution. That means generating new beta cells from, in my case, embryonic stem cells or pluripotent stem cells, IPS, induced pluripotent stem cells. Embryonic stem cells derive from the inner cell mass of the embryos, these are human embryos, and the inner cell mass has a very low methylation, and it's the situation in which that's an embryo of 5-7 days, in which the inner cell mass is in the best moment to have all the potency open to generate new cell lines. We have generated different cell lines, and we have cell lines generated in CABIMER that are healthy cell lines or cell lines that come from different diseases that could be used to see the history of the disease. Also, we have induced pluripotent stem cells using different variations of the Yamanaka method. And basically, in vitro differentiation responds to several aspects. Epigenetics, so you have to know a little bit about how to drive the cells into a new fate. That means basically giving orders that modulate the gene expression. So you have to know a little bit about epigenetics. So we did some work on epigenetics in collaboration with Manel Esteller and Mario Fraga on the epigenetics of these cells. And that also, you have to enter into the cancer history because it's, embryonic stem cells and cancer are very similar. Please take care. Very similar in the sense that they proliferate and differentiate. The point is that cancer cells do that in a pathological way. Embryonic stem cells do that in a physiological way. But they share certain properties, and we could see that there are a group of about 800 genes in which methylation does disappear in normal tissues, but not in cancer cells, and that somehow will reinforce the idea of the aberrant methylation of cells, of cancer genes, so that genes that have not lost the methylation of cytosine, and that's the reason why they proliferate and differentiate, and also a basis for epigenetic treatments. We have other markers, other molecules that we could use in the process of differentiation, but that's basically epigenetics. I've been an electrophysiologist for many, many years, and I couldn't resist the temptation of doing some electrophysiology, and we have described, there are very few papers describing the electrophysiological properties of human and mouse embryonic stem cells, so that gives us other strategies to force the differentiation into a particular fate. So taking all this information, we develop new treatments, and the summary will be this part of the slides in which, in blue, they are basically the mouse differentiation strategies. Most of them were from my group. My group was the first to describe a strategy. And in pink are the human differentiation strategy. I will concentrate basically in the group of Kevin Damour and the results from BioSite. BioSite is the company that has transplanted the first patients, and to my knowledge now there are 40 patients in the first trial, in the first pilot study, in which these patients are receiving, at least there were 13, I don't know how many there are exactly now, but there are 13 patients that have received the cells derived from embryonic stem cells, so it's an allogenic transplantation, so they will need certain immunosuppression inside a device that is located or transplanted below the skin. And there are recent publications that probably links this history of differentiation process with the history of Pedro Herrera, in which it has been observed that depending on the state of the host, the progenitor goes into alpha or into beta cell. And that's relevant because the patients of the California group are receiving progenitors of beta cells. So depending on the host, these progenitors could go into alpha or beta. If the host has no thyroid problems, probably will go into beta cells. But if it has a problem in the thyroid, probably will go into another cell type. And that's the reason why we are working basically in maturation. So our aim is to get more mature cells, and that's basically to summarize the results with the mouse that was published more than 15 years ago. And obviously we could show that these insulin-producing cells derive from embryonic stem cells, normalize glucose in diabetic mice, but also we move into human. And basically, very important in my opinion is to establish the minimal criteria. Sorry for this slide, but I don't want to comment all the topics. Basically, we have to establish which are the minimal criteria to accept that a cell is a beta cell or an alpha cell or whatever you want, because when you do in vitro differentiation, you end with a cell. A single marker is never enough to characterize a cell, so you need a list of markers, of responses, of physiology in order to have the minimal criteria. And the summary is that in the mouse we could show that our different protocols that we developed accomplish for most of the minimal criteria. And in order to summarize, it's important, some of these slides, the response is dose-dependent. For example, in the case of the mouse, for 1 million cells, the effect is transient. For 5 million cells, the effect is permanent. And when you remove the graft, you have to go back into the diabetic condition, or we could show maturation of the cells. The cells gain more insulin after the transplantation into the host. The same has to be shown for insulin-secreting cells for human embryonic stem cells or IPS, induced pluripotent stem cells. We developed a protocol which is basically a mixture of protocols, and now we are in a condition to say that we have the CABIMER protocol, which is a combination of protocols. Some of the factors are contributed by our group, the nitric oxide effects, the sirtuin-1, the cyclopinin, the inhibition of the sonic headcox, the nicotinamide. These are publication for my group, but basically is how to divide the problem into 7 different steps and trying to see how can you go step by step, trying to reproduce the development of the islet. And in our case, it's important to reach a cell like this one, in which green is insulin, red is C-peptide, and the combination of insulin and the merging gives a yellow signal, and the content or the number of insulin-positive cells is higher than in the conventional method. So we have procedures which are quite good. And the next step, because I want to say how far we are from the situation of treatment of patients, now we have a tentative protocol, I will say, the CABIMER protocol, that in our opinion is better than the other protocols, but that's the opinion of everyone. And we have to reproduce everything in GMP conditions. So we have to go to clinical grade embryonic stem cells. We know how to produce new embryonic stem cells, but that inside the GMP facility, reproduce the protocol at least 3 times, and once you have the cell, you could apply for a treatment. Recently, we have received a small amount of money from the FIS to do this kind of work and to reach this clinical grade insulin-producing human cells that could be used for a proposal of a clinical trial. Basically, we use the developmental approach, so we learn from the people who are doing development, Annabelle Rojas or Benoit Gauthier, and we concentrate in center factors, so we are learning different strategies. That's basically the way. And also the small molecule approach means using molecules that include forming embryo bodies, forming aggregates, what we call also mini islets, and using different strategies to reach this kind of cells. And we have one publication and another one that will come. And the next one is about, even forget even more mature cells, the strategy has been to use small RNAs. The initial idea was that if a human islet, let's say an adult beta cell, have a package of small RNAs, if we take these small RNAs and put all these small RNAs into an embryonic stem cells, we should be able to transform directly the embryonic stem cells into a beta cell. We did that. It didn't work. But now the strategy has been to first detect which is the mRNA, the microarray of the embryonic stem cells, the microarray of the islet, see how microarrays are moving, concentrate in a list of factors, and when we combine 2 or 3 of these factors, we get more mature beta cells. So that's basically the current situation, and that the next step will be to have these clinical grade insulin-producing cells. You could think as other thing that this problem has been solved. I mean, if the people of Biaside, California, which are basically Janssen, is the company that owns Biaside and also owns Vitalogic, another company that merged with Biaside, they are in a phase 1 clinical trial with all the support of the Juvenile Diabetes Research Foundation, etc., the California Institute of Regenerative Medicine. What are we doing here in Spain? And should I remind the history of Carrasco y Formiguera, who was a young doctor from Barcelona who went to Boston, learned how to purify insulin, and 6 months later established purification of insulin in Barcelona, and insulin was used in this city. Just 1 month later, it was used in Boston. So the history of insulin is almost 100 years, and we are still generating new insulins. I think the history of insulin-producing cells from pluripotent stem cells could last for decades. And we have new aims who have better beta cells that could better adapt to the needs of the patient. So that's the reason why we're still keeping the same. The third topic is about the regeneration, and that will be very short because basically refers to the work of Benoit Gauthier from my department. And although this paper is a paper that in which I was one of the authors, basically this is his work, and he has, or he defends that PAX4 positive cells in adult islet represent a population, or there is a subpopulation in the PAX4 positive cells which are still expandable. These are the cells that do expand during pregnancy in the mouse, but also Benoit has filed one patent that has more molecules on the process by which they could activate the PAX4, and his idea is to promote beta cell expansion from the adult islet. And if that is the case, that will be much better than any other approach because we could modulate the number of active beta cells. We still will need the immunomodulation and the protection from the antibody attack, but this is one of the approaches which is followed in my department, and that's basically the work of Benoit Gauthier. And I will dedicate the last part of my presentation to the use of cells, and basically I will show data from one clinical trial, although we have done several of them. And it's the use of cells in the treatment of diabetic complications. And I will concentrate in the critical ischemia of the limbs, that, as you know, in the diabetic patients is different from the peripheral artery. This is the end stage of the peripheral artery disease. But in the case of non-diabetic patients, most of the time you can revascularize using surgical methods. But in the case of diabetes, the damage is mostly distributed in small vessels, so that means that you don't have a surgical method to solve that, and amputation is one of the ends in most of the cases, and that's the treatment. And together with amputation, you have a patient that most probably will have problems with the kidney, problems with the heart, stroke, etc. So they are in a very bad condition. The initial idea that seems to be true, but there was an hypothesis that we knew that in the case of diabetic patients, there is a low number of endothelial progenitors in the peripheral blood. So we say, if the endothelial progenitors are in the bone marrow, why we don't take the bone marrow mononuclear cells and inject these cells into the artery and see whether we can generate that? So that was the proposal. We even published the proposal, and we begin the trial. Basically, I will show data from another trial. This trial was published a few years ago, and that was a pilot study with no control group in which we used mononuclear cells, very easily isolated from the bone marrow, and injected through the artery of the patient, the same patient, of the donor. That could be done in a single process in the operating theater. And as I showed, we didn't have a control group, but that was necessary for several reasons. One of the reasons is that there was a previous publication in Lancet in which the mononuclear cells were used in peripheral artery disease, but they exclude diabetic patients. So they exclude any patient that has glycosylated hemoglobin. And we include patients that have very high glycosylated hemoglobin, and the summary is that the system works even for patients that have a high glycosylated hemoglobin. We also validated a new blind system to measure the formation of new vessels. That basically is a computer program, a software that has been used to measure the dendrites and the axons of the neurons, but we adapted the software to this formation of new vessels. That means the number of branching segments, diameter, etc. But that was relevant because we could use the system in the next trial. The next was that's the trial that has been finished and hasn't been published, in which we have 3 arms. One arm is a control arm in which they receive the conventional treatment, and in the other 2 arms, initially there were 3 arms, but I will comment why we decided not to put the third one, with an escalating dose. The lower dose was 500,000 of cells per kilogram. The next dose was 1 million of cells per kilogram. And in this second arm, we have 2 patients with an adverse reaction. And that's very important because the adverse reaction that we had was microthrombosis. Microthrombosis is the last thing you will like it to have in these patients because these patients have a gangrene. And that was completely in contrast with the published data, because the published data says that mesenchymal stromal cells are fibrinolytic. And it's true. Healthy mesenchymal stromal cells in vitro are fibrinolytic. But mesenchymal stromal cells from a diabetic patients could do the opposite. So that is a good example by which we communicate adverse reactions to the regulatory agency, we stop the trial, go to the bench again, and try to find the reason why. And once we found the reason, we could come back into the trial, and I will show the results at the end. But at least I want to show you some unpublished results about the use of adipose-derived mesenchymal stem cells in this end stage. This is an open, multicentric, was done in 2 centers at the same time, prospective, and randomized. So the patients were randomized before being in one or another group. And the process of expansion is quite simple. As you know, basically we take the sample, 40, 50 grams of subcutaneous adipose tissue, transport into the GMP facilities of CABIMER, and in CABIMER we do the expansion, and after we do the treatment. And the expansion is something that we dominate because we have prepared more than 100 doses for different treatments. The primary endpoint was safety and feasibility. This phase 1 is safety and feasibility, but we also could say that it's safe. I will comment about the adverse reaction and how to control the adverse reaction. It's feasible, and also I will show some data of efficacy, because obviously these patients were followed, and in terms of efficacy, here you can see how these are samples of the videos of the angiography. That's the baseline situation. That's after 6 months of the dose receivement, and that's after 12 months of a patient that received 1 million per kilogram. And one of the first observations is visually you see this increase in the vessel formation that after we will analyze that with this metamorph method. And obviously I have not an answer of which kind of mechanism makes a single dose to generate effect 6 months later and 12 months later. So something changes, but that's another question that I cannot answer now. Obviously the criteria, the quantity, all the, well, I put two of these criteria, vascular density or branching, extension. There are a list of long of criteria, but you could see that as seen visually, the system, this blind system to analyze also show the increase in the density or the increase in the branching, etc. And basically in 35%, the increase was at 35%. The effect of in the ulcer was also very clear. This is a dry ulcer. That's, it's not bleeding. And when you see this pink granulomatous tissue scar formation, that is very good for these patients because that will bleed. And basically, if you go from, these are the highest complications, and when you go to the normal situation after 12 months, most of the patients go into a stage A0, A1, which is close to the normal situation. Also with analogic scales of pain, the improvement is quite clear in the case of the higher doses. Or what is more important for these patients, the Rutherford-Becker classification, which mixes a long list of factors, and basically grade 3 or category 4, 5, 6 are the most complicated situation, and that will be 0. And if summarizing all the results, you could see how this scale goes from a situation in which the control patients also ameliorate a little bit, but nothing that you can compare with the case of the patients treated with a higher dose or with a Miller dose. And when you go to this factor, you see that most of the patients go from this situation into this situation. That's the idea. Also for the oxygen, the transcutaneous oxygen pressure, that's quite difficult to have good data, but at least we could see some improvement, etc., etc. And the last comment is about this adverse reaction and how we solve that. The point was pro-thrombotic events. So we take the cells from the diabetic patients and analyze the expression of a series of genes and factors related with coagulation and fibrinolysis. And at least we found, we have more data now, but PI1 and TPA, which are directly related with the process of fibrinolysis, PI1 will be against, TPA will be in favor, but go in the opposite sense. And basically, when you take mesenchymal stem cells from diabetic patients and cross these cells with the sera from diabetic patients, the answer of increase in PI1, decrease in TPA, or decrease in dimer formation is higher. And that we published that in diabetes, also went back into the cells, and now these are also published data. We have assisted to rescue the phenotype. So that's a list of the cells that come from these patients have a lower proliferation, a lower migration. They express also tissue factor, etc. And now we have a way to rescue the healthy phenotype, treating the cells with PDGNF. Basically, I think that's the summary final. And the summary will be, we are in clinical trials in the case of peripheral artery disease. I have not shown any result of the same protocol used with non-diabetic patients, but I could say that the results are quite similar. That I selected this group of this trial on diabetic patients. In that case, we are in clinical trials. In phase 1, we have to move into phase 2, and we have the protocols, we have the cell production, the logistic prepare. In the case of immunomodulation, we are close to make the proposal of a trial for a short group, a pilot study with 10, 12 patients, recently diagnosed. I will say non-randomized, but that's something that we could discuss, because basically the idea is if we could protect and for a follow-up of 1 year, see whether C-peptide is maintained. In the other 2 cases, in one case we have still to generate the clinical grade embryonic stem cells, and in the other there is a new spin-off of CABIMER that will develop the patents and commercialize these tentative molecules that induce regeneration, expanding PAX4 cells. Thank you very much for your attention, and I think this is the last slide. Most of the trials were done in the Macarena together with San Lazarus. San Lazarus Hospital is considered to be probably the eldest in Europe, was founded by Alphonse X, and the message is that when you have the cells, you could apply the cells almost in any hospital, because the procedure is quite simple. And that's my original lab. In 2004, I established a new lab in the University Pablo Olavide. Now is the new center of CABIMER with more than 200 people, but that was the embryo. the original embryo of CABIMER. Thank you very much for your attention.